Hi, my name is Sabrina Templo. This is Bio 225. Week 4 today is June 2nd, 2020. And today I'll be doing the biochemical testing for microbial identification. And it's going to be the methanol red and Vogues-Proskauer test. And it's going to be on the active cultures for E. coli and S. epidermidis. So I have my MRVP broth, and I already labeled it as E. coli. And I have my E. coli culture. And in this one, I'll be using this bacteria to demonstrate my lab experiment. So I have my pipette is sitting in alcohol. So I'm going to let this dry. And what I'm going to do is I'm going to collect my active culture, some of it, from the E. coli broth. So first we're going to do sterilization. So we're going to sterilize the lid by passing it over the flame. And then I'm going to collect some of the bacteria. I just want to make sure it's dry first. And then I'm going to collect some of the broth. And then I'm going to recap, but before I recap, I'm going to sterilize again. And then I'm going to get my MRVP broth, and then I'm going to sterilize it. And then I'm going to put in at least 4 drops of the E. coli into the broth. So 1, 2, 3, 4. And then I'm going to dispose of this in the 10% bleach solution. And then I'm going to sterilize the lid. Okay, and then now I'll be placing this in the incubation location for 48 hours. And I will wait to see if there's any growth in my broth. And if there is growth, then I'll be able to tell by the turbidity. There'll be some sort of growth or floating or turbidity. If there is no growth, then I'll be having to incubate for an additional 24 hours. The test continues. So what I'm going to do is I'm going to have 2 tubes. I'm not going to be able to do this right now, but I have to do it after the results. So I have 2 tubes of each. I will sterilize them by soaking in alcohol. I have 2 for S. epidermidis and 2 for E. coli. So what I'm going to do, let me just put this over here. It's easier for me to see. So the first step is that I'm going to have my methyl red. This is a reagent. I'm going to use this, and I'm going to put it into 2 test tubes. So 1 of each bacteria, so 1 E. coli and 1 S. epidermidis. And I'm going to drop, well, before that, I'm going to have the MRVP broth, and I'm going to take half and put it into 1 tube, and the other half is going to go into the other tube of the E. coli. And the same with the S. epidermidis. So, and then the first 2 test tubes, I'm gonna drop 7 drops of the methyl red reagent, And if it's positive, then the broth will should show a surface of red at the top of the broth, so be positive. Then for the other 2 test tubes, the other half of the MRVP of the E. coli and the other half of the S. epidermidis, I'm going to add first the Barrett's A reagent. And I'm going to put 12 drops in each test tube. After that, I will swirl them, swirl the test tubes, and then I will add 4 drops of the Barrett's B reagent. And with that, I would, again, that's 4 drops to each test tube. And I'll also swirl the test tubes around and let them sit about 30 minutes in the test rack. And once I have the results, then I would observe the color and put it into my report. And that concludes my lab experiment. Thank you.